Halophiles, or salt-loving microorganisms, thrive in environments where most life cannot. From low-salt niches to saturated brines, low halophile have 1 to 5% salt, moderate halophile 5 to 20% salt, extreme halophile 20 to 30% or salt. Halophiles lives in saline environment. They maintain osmotic balance by accumulating compatible solutes or by adopting the salt-in strategy. High salt concentrations cause osmotic pressure that destabilize membrane and dehydrate cell and denature protein. But halophiles overcome this by modifying the lipid composition, protein interactions, and ion balance within their membranes. Let's move on to low halophile. Low halophiles, also called slight halophiles, are microorganisms that require a small amount of salt. Instead of using the salt-in strategy, like extreme halophiles, low halophiles accumulate organic osmoprotectants inside the cytoplasm to prevent water loss. These solutes are non-toxic and help maintain cell turgor and enzyme stability. Their membranes are similar to those of ordinary bacteria, primarily composed of phosphatidylethanolamine and phosphatidylglycerol. To resist osmotic stress, they accumulate compatible solutes such as trihulose and glycine betaine. These solutes stabilize membranes and proteins without altering the membrane structure itself. However, when salt concentrations rise significantly, the bilayer tightens, proteins misfold, and the membrane buckles, explaining why low halophiles cannot withstand brine. Their membranes lose integrity, and the cells cannot survive. Now move on, low halophile. Hello, I am moderate halophile. Moderate halophiles occupy saltier habitats, such as oceans and saline lakes. They do not use the salt-in strategy like extreme halophiles. Instead, they accumulate compatible solutes, osmoprotectants such as ectwin, glycine betaine, proline, trihulose. Some moderate halophiles produce light yellow or orange pigments, carotenoids, for UV protection. Their membranes are enriched with negatively charged lipids like cardiolipin, creating electrostatic stability. Unsaturated fatty acids increase fluidity, allowing the bilayer to flex and adjust even under osmotic stress. Some moderate halophiles combine solute strategies with selective ion transport, stabilizing membranes in fluctuating salinity. But in saturated brine, even these adaptive movements are overwhelmed. The bilayer ruptures and survival ends. Let's move on to extreme halophile. Extreme halophiles live in hypersaline environments. Their proteins are rich in acidic amino acids. Extreme halophiles, mostly archaea, rely on membranes unlike any others. Ether-linked isoprenoid lipids form exceptionally stable bilayers, and in some, monolayers that behave as a single sheet of protection. Their membrane proteins, rich in acidic residues, fold only in the presence of salt ions. The cell interior mirrors the outside. Potassium chloride accumulates, balancing osmotic pressure through the salt-in strategy. Pigments such as bacteriorhodopsin embed into the membrane, stabilizing its structure and filtering ultraviolet light. This is how these three halophile adapt themselves.